Hello, this is chapter 5 of the Brain Resource Creation Workshop, which focuses on thinking, planning, and language. We'd like to extend a warm welcome to everyone watching this video right now, and we thank you for your interest in this workshop. This presentation is brought to you by the Brain Resource Creation Department of Simply Neuroscience. It has been created by Natalia Martin, Melanie Hernandez, Nathaniel Dodson, and edited by Natalia Martin. Today's workshop is being presented by Melanie and Nathaniel.
This section of the presentation will focus on thinking and planning, specifically on how we think. The brain uses multiple types of memories and systems to perceive, respond to, and interpret thoughts or events which have occurred. Two key components of this process are concept subs and semantic memory, which we will explain in the following slides.
Semantic memory refers to the long-term memory of the meanings of concepts like object, date, people, places, etc. gained through experience. It's basic fact that we are usually not actively thinking about, but can recall pretty much immediately if requested to. For example, most of us know the dates of our birthdays. Semantic memory is constructed of concept cells, which we will talk about in the next slide. Research suggests that the specific cortical networks are specialized for processing certain types of information. This has been demonstrated by the fMRI scans, which show different brain regions firing in response to different information categories.
Concept cells are groups of neurons found in the temporal lobe that represent a sensory concept in memory, such as a smell, like garlic, or sight, like rainbow, and fire when an individual encounters these stimuli. They are responsible for the associations and transitions between related concepts and are thought to lead to episodic memories, or memories of personal experiences, and the flow of consciousness. The prefrontal cortex is also involved in higher cognitive functions. So let's talk about that.
Now, this part of the presentation will explore decision making. Decision making is the cognitive process of choosing between alternatives. This could range from clear-cut decisions, such as ordering food, to more complex decisions like selecting a mate. Our brain weighs values and rules to make predictions and plan accordingly. Our decision making also can be affected by strong emotional connection and motivation. This top-down mechanism involves the amygdala projecting information to the nucleus accumbens.
This section of the presentation will focus on regions in the brain involved with cognition. The visual word form area deals with the ability to recognize written letters and words. This region is also located in the fusiform gyrus. Moving on, we're now going to talk about the part of the brain that deals with visual recognition and reading. Being located in the occipital cortex and parental cortex, this region deals with the ability to identify symbols and connect it to a concept.
Now we move on to talk about visual disorders. The first language disorder we will cover today is agnosia. Agnosia is the inability to recognize and identify objects, people, or sounds using one or more of the senses despite normally functioning senses. An example could be that a patient with agnosia cannot identify a cup by sight, but they can state its color and identify it by touch. Agnosia is caused by damage to the pathways which connect the primary sensory processing areas, such as the posterior parietal cortex, occipitotemporal regions, etc. Such damage could include strokes, tumors, infections, dementia, head injury, developmental disorders, or neurological conditions. Agnosia cannot be cured, but can be treated with rehabilitation techniques.
The second visual disorder we will cover is prosopagnosia, also known as face blindness. You will break apart the word. It contains the word agnosia, which means that prosopagnosia is a type of agnosia. Prosopagnosia is the ability to recognize and identify faces. This disorder is caused by bilateral damage to the fusiform face area, which is located on the underside of the temporal lobe. Prosopagnosia differs in severity, as some people with the disorder may have difficulty recognizing a familiar face, while others may not be able to even identify or distinguish between a face and an object. Prosopagnosia cannot be cured, but it could be aided by voice recognition technology and using clothing as clues to help identify familiar people. Did you know that famous neuroscience Olivia Sacks had prosopagnosia?
Now on to the semantic system. Semantic system is divided into three regions in the brain that represent words. There is the prefrontal lobe, the temporal lobe, and lastly, the posterior lobe. It is important to know that the semantic system is also vast, which is that's why we can communicate.
It is also important to note where we control the majority of our cognitive function. This region is the prefrontal cortex. It is where most complex brain processes occur. It watches all over brain regions to make sure they are doing their job and intersects with all the posterior cortical areas. Pretty impressive, right? Unsurprisingly, it is the last region to finish developing, and it usually reaches where it needs to be to perform optimally by adulthood.
There are three primary executive functions that help us live a rather adaptable and drama-free life. Firstly, there is inhibition. Inhibition ensures that one does not express inappropriate behavior. Secondly, there is working memory, which helps some to remember a rule or code of conduct to help determine how they will behave. Interestingly, this working memory will vary between individuals as it is dependent on the parietal region and the prefrontal cortex, as we discussed, important for higher cognitive function. Lastly, shifting allows one to change their behavior as the environment changes around them. For example, when you find out that you're running late, you may walk faster or take another route that you think is a shortcut. All of these executive functions take place in the prefrontal cortex and the frontal lobe.
And another cognitive process is mentalizing. Mentalizing is making sense of one's own thought processes and those of others. This takes place in the medial and lateral prefrontal cortex and is involved in empathy and the theory of mind, or understanding mental states of others. Another key aspect is mentalizing is a temporal junction, or the part of the brain that focuses on others' behaviors. In that aspect, mirror neurons are very similar as they also deal with focusing on other behaviors. Mirror neurons, though, differ in that they fire when watching someone do a task that would fire if they were doing the same thing. But it's also important to note these neurons are controversial in the neuroscience community as they are found in monkeys, but few studies have hard evidence that they exist in humans.
Before we continue to the next section of this workshop, we will discuss additional brain regions related to language. Broca's area is involved in the production of language, and a lesion to Broca's area would lead to an impaired or complete inability to produce spoken language, or Broca's aphasia. Vernick's area is involved in the comprehension of language. So people with Vernick's aphasia would experience a partial or complete inability to comprehend what is being said to them. For example, if they are asked a question such as, "How are you today?" they would be able to speak a sentence back, but it would be complete gibberish. The middle and inferior temporal regions help process the meaning of words. Lastly, the anterior temporal region is currently being studied due to its correlation for possible sentence level comprehension of language.
The most relevant language-associated gene for the purpose of this workshop is the FOXP2 gene. The FOXP2 is responsible for the development of speech and language in humans. Mutation in this gene result in a lack or insufficient protein production. The downstream effect causes the individual with FOXP2 gene mutation to have difficulty making mouth and jaw movements essential for speech. Notably, a study involving birds displayed that FOXP2 gene mutations negatively impact signaling to the dorsal stertum, which is essential for motor control, thereby causing difficulty to communicate to one another.
An additional type of language disorder related to difficulty understanding speech is pure word deafness. This language disorder typically occurs when there is damage to the superior temporal lobes in both hemispheres of the brain. Sufferers of this condition cannot understand spoken language. However, they understand written language without any difficulty.
Another researcher focused on language was Karl Vernick. He was a German neurologist who wrote about a woman who had fluent aphasia, meaning she could speak fluently, but her speech just made no sense. Eventually, an autopsy showed damage to her left temporal lobe.
One case study involved with language and thinking is the case of Louis Victor Lebrun. Louis Victor Lebrun, otherwise known as Tan, was a patient of Paul Broca in Bicester Hospital. He was nicknamed Tan as he could only say one word syllable. Pierre Braque found that Tan could understand language spoken to him and tried to respond, but could only utter the syllable Tan. Eventually, Paul Braque would perform an autopsy on Tan. The autopsy showed a lesion to a part of Lebrun's frontal lobe, the Braque's area. It was concluded that the cause was damage to the left frontal lobe, which causes non-fluent aphasia. The diagnosis was non-fluent aphasia, which is when speech production is impaired, but comprehension is basically intact.
This concludes Chapter 5 of the Brain Resource Creation Workshop centered on thinking, planning, and language. This workshop was brought to you by the Brain Resource Creation Department at Simply Neuroscience. We hope you enjoyed the workshop. Lastly, if you have any questions, you can email us at the emails on the slide. Stay tuned for more videos from us. Have a great day from the people of Simply Neuroscience.